All right, so it's my great pleasure to introduce our special speaker today, Dr. Cheng, and he's currently the Randolph Terry Distinguished Professor of Regenerative Medicine at UNC Chapel Hill and the NC State University. He's the director, and he directs his own lab, the Biotherapeutics Lab. He's also co-director of the NIH Comparative Molecular Medicine T32 training grant. I was told, in fact, he also have been taking a lot of university level administration positions, including as the executive director of the interdisciplinary scholarship in the office of provost. Ke has been, I guess, developing, in fact, therapeutic, either based on stem cells or exosomes for different, I guess, different disease. And I guess you will hear more today from his talk based on Ke's research excellence and achievements. And he has been elected as a fellow of International Association of Medical and Biological Engineering and the American Institute of Medical and Biological Engineering and the American Heart Association. So with that, I should also point out Ke led several international new drug applications obtained from FDA, and Ke is the founder of several very successful startup companies. All right, Ke, without further ado, and welcome. And
yeah, thank you. I assume people can hear me okay, both at Zoom and in the classroom. So thank you very much, Jianping, for this invitation. And this is my honor and pleasure to visit University of Michigan and Ann Arbor, particularly the Department of Mechanical Engineering. Talk to lots of great people today, and also some of them are, I would say, old friends like Jianping, and also Andy Putnam in BME, who I had the privilege to serve in the same study section. And also I saw some names, familiar names like Professor Peter Marr, really a leader in the biomaterials field. I remember when I was a graduate student reading Professor Marr's paper. So it's great to visit UM. This is actually my first visit to this great university and ever been to Ann Arbor. So as you can see the title, drug delivery. I think the original title is drug delivery for cardiac and lung repair. I changed a little bit to cellular and biomaterial because we, as a lab, we don't work on small molecule drugs. The drug that I try to define, the drug that we work on are more biologics, therapeutics, like either cell therapies or biomaterials. So I started my career as a, I would call myself cytotherapist, which is using cell therapy to repair cardiac and lung injuries, starting with like those first generation cells, such as bone marrow mononuclear cells. And then the field moved to the second generation with more tissue specific stem cells. And also I know Jianping and many other folks work on like iPS cells and ESL therapies for cardiac and lung injury. And then the lab in the last decade more moved towards like cellular biomimetics and also like EVs and exosomes are a big part of the lab right now. And also in combination with injectable, inhalable biomaterials, organ patch, and for target delivery purposes. So I started with this slide that's talking about a disease that we're interested in. It's called idiopathic pulmonary fibrosis, IPF. It's from this CT image. You can see the scarring, the honeycomb structure of this lung. The black is actually the viable tissue, and the white area is actually the fibrosis structure. And you will see that this is a very deadly disease with a 5-year survival rate, actually less than 30, around 30%. Actually, it's worse than many cancers. It's only actually lung cancer and pancreatic cancer are worse than this disease, but this disease is worse than many other cancers, right? So obviously, there's only 2 FDA approved drugs for that, and they neither stabilize the disease, reverse the disease or stabilize the disease. The only thing they do is to slow the declining of the lung function. So as a regenerative medicine, you aim at repairing the lungs using stem cells. One approach is using cells exogenous to the lung, such as MSCs. The problem is they have limited potency for lung repair. You certainly can derive those cells from bone marrow or fat tissue, but you have limited potency for lung repair. And then the other approach is using the cells who are resident to the lung, such as lung stem cells. But the problem you run into is difficult to purify and expand in the lab. So we kind of, in the last decade, we developed this called lung spheroid structures. They are not organoid. I think there's many of good folks here, like Jianping and Brendan and Claudia that have talked to yesterday working on organoid structure, but this is really a very simple spheroid structure that contains this is mixture cell population. You can derive either from a biopsy if you apply autologous application, or you can derive from a whole donor lung for allogenic applications. They express both epithelial stem cell markers and mesenchymal stem cell markers. We did a proof of concept study in 2015 by injecting those cells into a mouse model of pulmonary fibrosis, which is, in FDA's language, we call it POC study, which is proof of concept, preclinical study. And we obtained good efficacy and safety data by seeing these images here that reducing the fibrosis in the lung. And we went ahead to convert a lab to a GMP facility in the university submitted our phase 1 clinical trial with Dr. Jason Lobo, a lung biologist, actually a pulmonologist, director of the lung transplantation and IPF clinic, as a PI in collaborating with the two universities. And also my startup company, we started this clinical trial. We call it a HOT IPF trial, human autologous lung stem cell transplantation for IPF, which you can see that you have this structural collapse in these IPF patients. We go through this, it's a bronchoscope, basically bronchoscopy to, it can be done in an outpatient clinic. With sedation, then brief sedation, and then you take these cells to my GMP lab, and we get both the stem cells, but also supporting cells, and then go to the second stage, which we call the lung spheroid, in a suspension culture to increase the stemness. In those suspension cultures, that the cells will self-aggregate into these miniature lung spheroid, and then we harvest them to get to the single cell population again, so that we can do IV injection in those patients with IPF. So the patients usually come to our clinic and to get their biopsy, and then we grow the cells. In about a month to 2 months, they can receive their cell therapy back. So it's autologous cell therapy, and so far we have done 9 or 10 patients, and with excellent safety. We will report efficacy data sometime this year. So this is a cell therapy, and as I alluded that my lab also work on exosomes. So why we're interested in from cells to cell derivatives like acellular agents? The reason is stem cell. We know that adult stem cell, including MSCs and LSCs, like we're working on, they exert their benefits mostly from paracrine factors. Those paracrine factors entirely things are called secretome. One fraction of that is soluble product, which is like proteins and cytokines. And then you have these acellular, actually not acellular, the insoluble fraction with EVs, extracellular vesicles. The benefits they offer is less tumorigenicity, less immunogenicity, off the shelf, stable and longer shelf life. So before we work on EVs, actually a talented student in my lab, Tyler Allen, and in collaboration with Dr. Junnan Tang, a visiting scientist, a former visiting scientist in my lab, they created this technology that we call it synthetic stem cells. Basically you're not using the cell, but you since you know that most of the benefits are from their secretion, so you take their secretome and package those secretome into a microparticle, and then you also do one step further, which is put the cell membrane on top of these particles, which is to increase the adhesion and also to preserve the cell binding molecules on these particles. So we call this cell mimicking microparticle, CMMP, which is a particle like inside out mimicking a real stem cells. And then we inject them into a mouse model of myocardial infarction. We did it's essentially the same thing, CMMP, but we call it a synthetic MSCs. It's essentially CMMP, the microparticles. And this time we do the MSC. As you can see from the circulation research paper here, their benefits in this particular cardiac injury model are very similar to genuine natural stem cells. So what's the purpose to do this synthetic cell is that you have this good stability and also off the shelf, because these synthetic particle, MSC particles, you can lyophilize it into powder, and whenever you need it, you can reconstitute into the resuspending PBS, which you probably never can do for real stem cells. You need to be carefully preserved and carefully characterized before you can apply them. Because so basically where this process we call the pharmacoengineering of stem cell therapy is to convert a living drug to a non-living drug to increase stability and convenience of off the shelf, right? Because you cannot go to a Walmart or CVS to tell the pharmacist that I need my stem cell from the shelf. It's never gonna happen. But maybe with this, there's still many steps in the future, but we are on that track that to pharmacoengineer a cell therapy to a non-living drug with convenience. So this is a total secretome, which is including EV. And I mentioned what extracellular vessels and exosomes, and I'm going to use these two terms interchangeably in my presentation. So what are exosomes? Exosomes are basically the smallest fraction of extracellular vesicles secreted by a lot of cells. Essentially, all type cells, they secrete EVs. And EVs are very important in cell-cell communication, right? So a donor cell secretes EV and uptake by a recipient cell. And then recipient cell, this uptake of EV will change the recipient cell's biological functions. So this is like a remote communication between two cells. They don't have to actually touch each other, right? It's just like we can use short message, text message to communicate. We don't have to stay in the same room, right? Same thing. So EVs are good cell-to-cell communication tools. So as I said, the lab is very big in EV research right now. We even started our own journal, Extracellular Vesicle, in collaboration with Elsevier, in collaboration with Professor Raghu Karuri at UT MD Anderson Cancer Center, and we have also this nice society, American Association of Extracellular Vesicles, that we're doing annual meetings every year. So last year was in California, and I think this year we're considering to have this meeting in the Boston or New York area. So hope whoever interested in EV can join us. So EVs, how do we use EVs? We can utilize EVs by usually two directions. One is natural EV, just taking the EVs naturally secreted by the stem cell, and then use those things as therapeutics. And the second direction is engineered EV, which means you take an EV from a generic cell line, but you load it with some drug, right? So you're using the EV as a drug delivery tool. You're not relying on the natural payload, but you have actually to load a payload, that's a protein drug or mRNA in the EV, but utilize the drug delivery application of that EV to deliver your drug, your mRNA, your protein, or your small molecule. So I will talk about the natural EV first. You remember that we started this trial using lung spheroid cells. And as the trial is going on, one of my former graduate students, Wen Ding, started interested whether we can, using the secretome or the exosome from these cells, maybe they have equivalent therapeutic benefits. Now, if you're using those tiny vesicles, one of the beauties that you no longer need an IV injection. You can do inhalation, which is even less invasive and can actually achieve repeated dosing, right? So basically, what she does is, to have this secretome and exosome from these cells, and just using a nebulizer to create this nebulization inhalation into those mouse, and the mouse actually, they reach out to that mist. We call it stem cell mist, right? Some students even joking that this is stem cell sauna. I doubt it, but anyway, so this is how we. Sometimes science is tedious work. You need to make some jokes to keep the excitement, right? So, but we actually get fabulous results. Is that by inhalation, this LSC secretome, you actually see a very good benefit of this. Is actually Ashcroft scoring is measuring fibrosis extent. It's actually very similar to the sham healthy animals, outperform the PBS group, and also with a trend better than MSC secretome. So basically, you don't need the cell; you just need the secretome or the vesicles secreted by the cell. Inhalation with repeated dosing can achieve a functional outcome in this particular mouse model of. Yeah, Brendan, you have a question?
Can you just tell us is this a single bleo dose model, and what's the time point if it is?
Yeah, it's a good question. So the bleomycin, lots of people ask me the question, bleomycin is not a good model, but people keep using that. Even the large companies like BI, Genentech, their drugs are still using. So we inject one dose of bleo, but we waited 10 days. Because if you inject your cells or inhale exosome right after bleo, you are really treating inflammation. You're not treating fibrosis. So we actually see, starting 10 days, inflammation is going down, and you see a quite big amount of fibrosis. So we actually published a paper to demonstrate the timeline. So we actually second animal at day 3, day 7, day 10, day 14. We actually day 10, day 14, because you also cannot wait too long. Because if you wait too long, it will self-resolve. This model is very different from human IPF. Human IPF never goes away, but this, it was self-resolving like day 20 or something. So you cannot treat very early, but also you cannot treat very late. So we choose day 10, and FDA accepted this day 10 as a good time point for fibrosis. Yeah.
For harvest or for starting your treatment?
Starting the treatment.
And then when do you harvest?
So we treat for 7 consecutive days. Yeah. We have done both day 10 and day 14 treatment starting, but we only do 7 doses, and then we harvest the tissue. So just as in a nutshell, the results is that, if you compare these, here we do some, you can show a bunch of histology, but the holy grail is whether it increase lung function, right? It protect lung function. So these are very tricky to do lung function, pulmonary lung function tests in those little rodents, but we have this equipment, and with some experience, we measure those lung function parameters, and we actually see that the exosome inhalation, actually you do see you reach the efficacy endpoint by, you see this in preservation of FEV, FVC ratio, and elastance, and also other measures which are clinically relevant and clinically used in the trials that for patients with IPF. So what we see here in a nutshell is that in this paper is that, we're doing clinical trial on this one, which is a cell, but we believe the benefits are from the secretion of the cells, including proteins and also extracellular vesicles, and also we are on the course of identifying which microRNAs and protein factors played as the functional ingredients in those, so we can further improve our therapy by enhancing those microRNAs to have like super exosomes, or even taking 1 or 2 of them and encapsulate in an LNP to form a sort of like get rid of the exosome and cell as a synthetic therapy for IPF. While we are doing a mechanistic study, we submit another IND for another startup company that I helped co-founded, and we actually get approved for this trial as well is inhalation of allogeneic, this time, lung sphere cell therapy for patients with ARDS, which is a acute lung injury. And we are actually about to kick off this second clinical trials. Now, so far we talked about acute lung injury and also IPF, but this cell therapy we do believe can be expanded in other indications, such as pulmonary arterial hypertension with heart failure with preserved ejection fraction. We actually see that 3 weeks of sorry, I'll just keep switching using mouse and laser point because I know the people on the Zoom cannot see my laser points. So I was using mouse from this time point going on, is that you can see 3 weeks of exosome inhalation. You will actually see that so this is a tedious model, that 28 weeks of high-fat diet in those mice to create this model, almost a half a year to create this model of PAH and heart failure. And then we see actually 3 weeks of exosome inhalation reduce the PAH by about 50% and heart failure by about 30%, which is quite amazing. And we think we can, with more study, we can launch another trial for this particular cohort of patients. What about other disease like COPD, CF? And we also see good results that with patients with chronic obstructive pulmonary disease and cystic fibrosis, we actually see the inhalation of exosomes reduce the layer and the thickness of the smooth muscle layers for this, which is culprit in this particular disease. So these are all natural EV therapies, but as I mentioned in the beginning that one way and the whole field is try to use EVs to do drug delivery. As I get interviewed by this particular Nature Outlook issue, particularly for EV and exoRNA, what I said is that the very big direction is how to use extracellular vesicle to enhance drug delivery, like what you can do using EV that outperform, will these EVs outperform LNPs or liposome, because liposome and LNP has been popularly used for drug delivery. So one of my students asked this question, can lung-derived EVs efficiently deliver therapeutics to the lung? So Kristen is no longer a PhD candidate, but she already graduated. Kristen's work is inhale exosome and then compare exosome and liposome in the efficiency of deliver mRNA and protein drugs. So liposome is similar, to LNPs, not very the same, but they share similarity. They're synthetic nanoparticle. You can load mRNA and load actually protein. So what Kristen did is to load we didn't do any drug, but we do GFP encoded in mRNA and then IFP protein. So basically, after inhalation, if you see GFP signal in the lung, that's the translation of the mRNA. And if you see red signal, that's the protein. So you will see that it's a co-delivery simultaneously. And you will see that actually the lung exosome outperform the generic cell exosome, which is hex cell 293 exosome, and outperform liposome. If you see the in this IVIS ex vivo lung image, you will see the signal abundancy that in the lungs is using the lung-derived exosome is kind of the highest, reaches the highest retention and biodistribution in the lung. This is in agreement when we take the histology. We see that the liposome, most of them are remaining in the trachea. And then if you look at the tiny, the little bronchioles and parenchyma of the lung, you will see the exosome really outperformed the liposome in drug delivery, both mRNA and protein into the deeper lung structure. And actually you can see those images that here that this is light sheet. So you use clearance technology to clear the lung lipids, and you can actually see the particles, the exosomes were distributed through inhalation. They distributed to the deeper lung tissues in a 3D way. By rotating it, you actually can see their deposition. And actually, with all three methods, ex vivo imaging, light sheet imaging, And histology confirmed the superiority advantage of exosome deliver drugs over liposomes in this particular inhalation model. So Kristen go one step further, is because all we have shown so far is liquid, which is nebulization. It's very different from dry powder. So what Kristen did is whether we can lyophilize, make a dry powder of the exosome, because dry powder, most of the pharmaceutical drugs are in dry powder. They have longer stability than liquid form, right? So what Kristen did is she lyophilized the exosome into particles using AFM to confirm the stability after freezing, and then actually see that after a 28-day lyophilized in a room temperature, the exosome still can efficiently deliver mRNA and proteins in this particular model. You still have a nice expression of both mRNA and actually the delivery of protein in this particular model. We actually see this in the monkey study. Actually, interestingly, we see that in the bronchial, you see this signal, which has the inhaled exosome after 1 week. You still see the mRNA translation and also the proteins in the parenchyma, deeper lung tissues after 1 week of 1 dose of inhalation, which is kind of long-lasting than the typical LNP-mediated delivery of mRNA. So far has been biodistribution. So what are we going to use this technology? Right? You have demonstrated a superiority of exosome over LNP or liposome in delivery. So what you can do? So one thing we're doing is cancer therapy, which is IL-12. So IL-12, we know that is a very powerful cytokine that can recruit cytotoxic T cells to kill the tumor cell. So what we do is we load IL-12 mRNA into the exosome and then inhale in a mouse model of lung cancer. So the notion here is that the lung cancer cells will uptake those exosomes and then produce the mRNA will translate into proteins, and they were pumping out these IL-12 proteins, and these lung cancers will recruit the T cells and other immune cells to kill the lung cell. So IL-12 has been, and people have been shown using IL-12 mRNA loaded in LNP, including in Darren Irvine's group, using IL-12 LNP for tumor therapy. Now, actually, we compare the IL-12 exosome versus IL-12 liposome. We actually see, just in a nutshell, in this image, you can see IL-12 exosome this is a tumor burden, a tumor growth. You actually see that the outperform the liposome. And this black line is the PBS group. You will see really it has the highest ability to eradicate the tumor and actually cause the immune response to have the microenvironment to actually be anti-tumor instead of pro-tumor. So this is, I've shown so far, is the power of natural EVs, exosomes in lung regenerative therapy, and also a little bit of the engineered EVs in deliver drug as a better drug delivery tools to the lung. So as we know, after COVID came, the whole group was thinking what can we do as a group to do some research to contribute to the science and engineering of this whole field. So we also use, as I said, use nanovesicles and EVs for COVID therapeutics and vaccine, and also inhalable biomaterials for lung protection. So hopefully I will, in the last 15 minutes of, about 13 minutes of my talk, 45 minutes. So to talk a little bit of this. So as we know, spike protein, the SARS-CoV-2 virus utilizes the spike protein to bind to the ACE2 receptors on our lung cell. And through this key and rock interaction, they use this key to open the lock, which is a door. And then this virus can go to our lung cell and do all these bad things. So our hypothesis is that if it's like a decoy. So if you can making those nanovesicles or nanobubbles from our lung cells, for example, using the lung spheroid cells, the same cells that we're doing clinical trial, we know they have natural abundant ACE2 expression on those cell membrane. So if you make a nanobubbles from these cells, they should preserved all the ACE2 on the surface. Then you serve like a decoy to saturate the spike proteins on the virus, right? So imagine that they have all the keys, but they're using fake locks to saturate all the keys. They don't have the key to open the real lock, right? So this is done by Zheng Hua, and I think I still have some time, so I can actually play this video.
While there are many approaches to treating the symptoms of COVID-19, there are currently only 2 approved therapies for emergency use: monoclonal antibodies.
Never mind. The video is very, actually the sound is not very, maybe there's a sound system. So for the sake of time, I think I can just skip it. So basically what this is just the images that I distilled from the movie is that the one lung cell, the lung stem cell that we used in the clinical trial, right, can generate 11,000 bubbles. So it's a number game, and you dramatically increase the surface area, have a better chance, right, to these nanodecoys have ACE2, and they actually bind the SARS-CoV-2, the spike protein on SARS-CoV-2, and form these aggregates that can be more easily and rapidly cleared by macrophages. So this is a nano decoy cell therapy, and we did a monkey study that we give real SARS-CoV-2 virus in these monkeys. No, we didn't do any of these studies in our university. We don't have a monkey center. So this is all done in a CRO who has BS3 facility to do SARS-CoV-2 study. And we give real virus, and then in day 2, day 3, day 4, day 5, we give 4 doses of inhalation of nano decoy. And then we actually do the measurement, and we actually see what this nano decoy inhalation does is actually dramatically increase the speed of clear the virus and also protect the lung structure. You don't have this injury that you see in these control animals. You see the lung structures are gradually, greatly kind of preserved. So basically, this decoy is help clearing the virus more rapidly, decreasing viral load. And it is clinically proven decrease of viral load is so essential to determine it's mild COVID or it's severe COVID. So basically, this is something that we think it's beneficial for as a therapeutic to apply when a patient has confirmed high-risk patients with COVID diagnosis. So what else we can do with EVs is that we can also, because the EV extracellular vesicle, their structure is so much like a virus. So you can actually make something like a vaccine, like a viral, there's a virus-like particle, VLP. So in this paper, this Nature BME paper, what we actually, there's actually these 2 scientists that call for Operation Nasal Vaccine, because the nasal vaccine, why we're interested in inhalation vaccine is that the Moderna, Pfizer, and those kind of intramuscular vaccines, they're very good at promoting systemic immunity, but they're not very good at promoting mucosal immunity. And your mucus, starting from your nose to your lung, deeper structures are all layered with this mucus. And that's your first barrier, your first defender. So if you have high concentration of antibodies in those mucus layers, you will have a faster reaction after vaccination to the virus. So what we do is we decorate the using the exosome from the lung and decorate the RBD, which is receptor binding domain. It's a part of spike protein. So we use this RBD as the antigen to decorate the exosome. And so make this to trick the body system like this is a virus. And then the body will produce antibody, and particularly IgA, which is classified as a hallmarker of mucosal immunity instead of IgG, which is more like a systemic immunity. So we created this, and we see actually room temperature actually is stable for 3 months. Actually, for this vaccine, room temperature is stable for 3 months. I think it's great to know that it's important for our transportation system that if you can have a room temperature stable vaccine, right? And we actually showed that they do increase the IgA, the mucosa immunity indicator antibodies in both nasopharyngeal lavage fluid and bronchial alveolar lavage fluid. Actually, also not only antibodies, but also you have a cellular response. The cellular immunity gets promoted by this inhalation of exosome-based vaccine. We did this study in a hamster, this time not in monkey, but in hamster. And we give 2 doses of vaccinations first. And those hamsters, the reason you use hamster is these golden serum hamsters, they get sick from the SARS-CoV-2. If you're using mice, the human ACE2 is very different, so they don't get sick unless you use ACE2, humanized ACE2 transgenic mice. So we use this hamster model, and we actually see that 2 vaccinations actually help protect them, from infectious and also reduce the disease burden, both viral burden and disease burden. Okay, so these are exosome work. So in the last 7 minutes, I just keep eye on my time. I'd like to talk a little bit away from all the things I've shown. Stem cells, EVs are all derived from cells, right? But as I mentioned in the title, we also work on a little bit on biomaterials and inhalable biomaterials. So here is a biomaterial that we developed and tested for as a kind of antiviral and as a defender biomaterials for viruses. So as we know, even we know that there's a winding down of this COVID, but we still see people. It's a choice right now that people either wear masks or not wear masks, right? And certainly we know the mask is still a very effective way to protect you and your families from getting the virus. And but there's also limitation of the virus. For example, when you are exercising, right, you are doing this exhaling and inhaling, and the mask is a burden to you, right? Alan, you're wearing a mask right now. And sitting down here, no problem. But if I ask you to do a long like a marathon, you wearing the mask is a burden, right? Because exercise. And swimming, can you wear a mask when you're swimming? Very hard, right? You can soak your body in the, but you can never do a real serious swimming if you have the mask. Eating and drinking. I know that we created all these inventions, but that mask will not do any job if you create a hole. And I think our celebrity, our politician, creating all those ways you can wear a mask. That you can have a mask. Yes, I'm complying with the rules, but I want to breathe, right? So these are all these ways. So what we want to do is to see if we can create something like an invisible mask that you can inhale and let your alveolar structure wear a mask, but not you. So this is something that we create a material that inhalable. We call it a spherical hydrogel inhalation for enhanced lung defense with a fancy acronym we call it SHIELD. Is that you can inhale this. Basically, these are material that these are mostly gelatin spheres, and that once it's inhaled, it will absorb water and then form like a hydrogel layer and network that cover on top of the mucus, which is serve as a additional barrier that to prevent that the virus can goes to the mucus and actually penetrate the lung epithelial cells. And actually we can see that in this histology of these rodents, you see this red color is the hydrogel, form a very nice layer and coverage of this alveolar structure. And just in a nutshell, we did the monkey study. We actually see that this inhalation offers 8 hours of protection. That after inhalation of this 1 dose of this is a dry inhaler, so you can inhale this. It's a very cheaper material. Actually, you can and food grade, like generally considered safe material that you inhale this particle. It will form a hydrogel and offers 8 hours protection from being infected by SARS-CoV-2. We actually also tested this inhalation does not affect your lung function, so you still can breathe and normally, and even with some kind of activities and or exercise. And this is not only work for SARS-CoV-2. We have tried that for flu virus, and also try that for other applications like allergy, pollen allergies. A lot of people in America are allergic to pollen in pollen seasons. Not so much maybe in this area, but in my area, there's a lot of pine trees. And the pollens are really, if you go to the car, your car has a very thick layer of yellow powder pollen. And then people are coughing, sneezing. And I think this hydrogel, we're doing some study and showing actually preliminary efficacy that can prevent your, protect your airways from those kind of particles. And also air pollution. There's countries that you still have a lot of PM 2.5 pollutions, and I think this will offer that. I think this will not replace mask, but as a alternative or enhancement to the mask, to your N95 or to your surgical mask. And in certain, we're human beings. When we interact with people, we would like to see your face, right? Face-to-face communication sometimes means a lot. And you wear a mask, yes, we can wear a mask and interact, but I lost your facial expression when you wear a mask. So we made it very clear to the editor that this is not a replacement of mask. We still encourage people wear a mask, but this could be a good alternative. So I think I will not have time to, my lab also work on cardiac stuff, but for the sake of time, I will not spend too much time on the cardiac stuff. Just give a very quick overview is that we do cardiac patch. We do cardiac patch work basically using this decellularized ECM and with the same synthetic materials as cells that I introduced at the beginning of the talk as a kind of a regenerative cardiac band-aid to treat myocardial infarction, as this all published paper that you can search. And we did pick studies to show that they are quite pro-regenerative or pro-reparative. And also we collaborating with our previous colleague who adding microneedles into those materials to create and also we're creating those devices that can insert it into the pericardial space to pumping these pro-regenerative materials. So these are all some of the work that we do, therapeutic work, hydrogels, injecting hydrogels into the pericardial space, and then release exosomes. This is just some videos I will not show. Basically, you can see here that we can inject the hydrogel that be able to sustain, deliver exosomes into the pig model of myocardial infarction. Very excited about this work. We think we can translate this work as well because in human beings, we can inject those hydrogels into the pericardial space with a very minimal invasiveness. This is just a fluoroscope guided intrapericardial injection with local sedation. Without even general anesthesia, you can do this injection to the heart for post-MI patients. We are on the course of getting IND approval or clinical trial approval in this space as well. And also, I like in the last year to show that in exosome device combination, what we do is we put the exosome on as a coating the exosome at the layer of the stents, coronary stents, and we think this will do two ways of work. Is that the exosome will render the stents more biocompatible. As you can see, the exosome coating on the stent make it a less foreign body reaction because it's like a camouflage. But the stent will also help the exosome to deliver to the upstream of the injury and then have kind of those therapeutic benefits. So just in a summary that I said, my lab works on cardiac and lung diseases. We started our work with cell therapy, and then now more and more using like those acellular components such as exosomes and inhalable and injectable biomaterials. With also the cardiac work, I unfortunately will not be able to show in a detail, but what we do is really to develop targeted and minimal invasive delivery for therapeutics to the heart is just a few slides that I showed you. It's also a big chunk of my lab, but if you're interested, you can talk to me or just read the recent papers from our group. So with this, I'd like to thank all the groups who did the work and also funding agencies. And yeah, happy to take any questions. Thank you very much.
impressive amount of work. I call it an impactful. I was going to make some comments in the fact that I remember Kurt was mentioning, given the presentation is so clear, Kurt mentioned to me his lab runs the biggest animal operation on campus.
Yeah, we do,
Obviously,
yeah.
very not surprising. I was also thinking about given the shell that you were talking about, still possible. I think it would be most useful for certain politicians who are refusing to wear masks.
I hope so.
One side for actions. All right. Okay, all right. Yeah, I just want to add some comments.
Thanks for the comment. Yep.
Yeah.
I have a question. Yeah, great, very impressive work. I have questions about your earlier, like the synthetic stem cells with
Yeah.
cell membrane microparticles. So you said basically you coat the particle with the
Cell membrane. Yeah.
And you can lyophilize them and dry them, and that retains activity after you constitute. Do you have any idea how that happens? And I guess also curious, do you have any idea what's the mechanism of protection? Like what is it on the synthetic cell or the stem cell membrane?
Yeah,
that
yeah,
actually operates?
yeah. That's a great question. So
It's not relying on the internal stem cell machinery, but rather the membrane.
yeah, that's a great question. So the reason we put the membrane on the particle, as I said, is probably twofold. One is to, if it's just it's a plain or naked PLGA particle, they have hard time to anchor to the tissue after injection. So you have to see a lot of loss. When you have this membrane, what we see is that it has a better retention in the injection site. So basically, I think some of the binding molecules of the on the membrane help them. Yeah, but it's on top of that. I think it's more than that. Is that we have seen that with that membrane, you have better therapeutic benefits. So we compare that without the membrane, you have less therapeutic benefits. The reason I think is that somehow the stem cell membrane, there's some ligands on the membrane, when it's in close contact with the injured cells, is on top of the secretion, it triggers some sort of signaling in the recipient cell. And I think beta-1, integrin beta-1 is one of them that we did the loss of function studies. If we block beta-1, then it diminishes the benefits of the cell membrane. So that's answering your second question, which is why you need the membrane. And the first question in terms of stability of the membrane through this lyophilizing process is certainly, we're still under optimization. I think the lyophilization process is it is a harsh process.
Yeah.
It has actually has we are still optimizing that. How do we, I think you will see some fragmentations. They are not as shown as nice as the cartoon, like it is single nice layer. I think it's never that perfect, but at least I think some decoration of those ligands is still beneficial to the particle itself. Yeah.
Very cool.
Yeah.
Thanks. Any other questions? Cheng.
Yeah, the talk was really impressive. Thank you. And I have 3 questions, if it's okay.
Yeah. Okay.
First, for the exosomes, if I take somebody else's exosome, would that cause an immune response?
Mhm.
And second is that for the gelatin coating, how do you make sure that it doesn't go all the way down to our alveoli to stay in the mucosal layer?
Mhm.
The third is that if you use a gelatin mask, do you see any behavioral difference in the monkeys? Maybe they freeze harder or
Well, you use it.
because
Well, the third question is if you're using gelatin max, you said.
the gelatin like
Okay.
inhalation.
Oh yeah.
If you do that in the monkeys,
Mhm.
do they show any behavioral differences?
Okay. Cool. Yeah. Oh, gosh. Yeah. Great question. So 3 questions. The first question is repeat the question since I think the Zoom people may not hear. The first question is, can you use allergenic EVs without concerning of immunogenicity? The field has some consensus that yes, you can use allergenic EVs. Their immunogenicity is less than you use the real allergenic cells. We are still on the course of identifying the reasons, but it's certainly, the EVs, you still have some of the like the molecules, like MHC molecules, but the abundance of these molecules compared to the cell is different. So in our hands and in other people's hands as well, you can even use xenogenic EVs in immunocompetent mice, and you don't see much rejections and immunogenicity. You would think, this is something that it's different, but it's a cell secretion. Certainly, the degree of rejection and immunogenicity is very different. It's a cell secretion versus a cell itself. If you give cell itself, you definitely will have some rejections and immunogenicity. So that's to your first question. The second question is, do we worry this gelatin microsphere inhalation go down all the way down to the alveoli space, and then from the bronchial and to the alveoli structures, and to actually have some detrimental effects, right? So the outcome we see from the lung function study, toxicity study, we didn't see any of them. And I think the reason is that you can optimize the size of your particle, because the size of the particle will determine how deeper they can go, right? If it's, we know that usually if you want to go to the deeper round, you need to be less than 10 micron, but not too small. If it's too small, it will definitely go to, like exosomes in nanoscale, they will go to the alveoli space. So far we're using something like around 1 to 5 microns, which is like in the, they will actually cover some of them will go to the upper respiratory tract, but some of them will go to the lower respiratory tract. But we don't think many of them go to the deeper, like those alveolar structures. But it's a good consensus that particle is important. But we can certainly do more study to give let's say a 400 nanometer microscope, because we can titrate the size and see if it actually causes toxicities. In monkey studies, we don't see behavioral changes after this inhalation. We don't see behavioral changes. Thank you. I think so, because the gelatin, there's actually a company making, they're not using gelatin, they're using Cerasus, kind of LPMC, which is a kind of a synthetic plant-derived kind of fiber kind of particle for the nasal inhalation to prevent from actually their indication is allergy protection, which is inhale the particle. Yeah, so gelatin is kind of like animal derived, but the other part is the counterpart is the plant derived particle, and they use that for, it's FDA approved, I think, for allergy protection. Yeah, particles as well. Yeah.
Thank you. Any other questions? No, please go ahead.
Thank you for your talk. Yeah, I wondered when you use EV for the lung therapy, so have you ever seen the unexpected effect or bad effect to the other tissues about the piece or
Okay. So the question is, when we use EV therapy, do we see any other unexpected effects in other organs? So we don't see toxicity effects in other organs, right? So when we do a FDA application, we submit the data for major toxicity events in other organs. We don't see any of them. Whether they go to other organs, yes, definitely. Even by inhalation, they will still go through the alveolar blood barrier, ABB, cross the barrier to go to the blood, and then you end up in the liver. So we see the EVs in the liver as well. Actually, we see the EVs in the spleen, in the blood. Actually, some of them we see them in the brain by inhalation. They go to the brain as well.
Very good.
Yes, small portion of them end up in the brain. I think they have a way to cross the BBB, maybe. There's people actually using EV to deliver drug to the brain for stroke or Alzheimer kind of application using EVs. So we do see them go there, but because EV is a natural stuff, we don't see much, these toxicity effects that you're worrying. Yeah.
We have to wrap up. It turns out we have an FBI information session.
Yeah.
FBI agent.
Yeah. Okay, cool.
But with the cell delivery, did you guys look for engraftment directly in the lung? Because you mentioned IV injection.
Yeah.
The bigger question is, do you know, could it just be exosomes being produced inside the body once you engraft cells somewhere? Like, do you have a sense for whether exosomes are mostly the benefit of the cells, or are there other things that cells could be doing upon engraftment?
Yeah. So engraftment, we actually see after IV injection, we do see engraftment in the lung. They reside in the lung, but they don't permanently engraft. So that makes us believe that the paracrine effects still play an important role in this type of model, because they do not permanently engraft and become a part of the permanent lung structure. They do have some differentiation because they are epithelial stem cell in nature. But if we do a GFP or human nuclear counting, after 3 weeks or 4 weeks, these cells are all gone. So we believe that the paracrine effect is still the major players in this type of model. Yeah. Okay.
I have 2 questions. In fact, let's wrap up 2 minutes. So in fact, I understand. My understanding, I have very limited knowledge about drug delivery system, but it seems like your EV delivery system, you really compare this, your delivery system versus liposome delivery system.
Liposome, yeah.
I'm wondering, I understand liposome is a synthetic model,
Mhm.
and people just, they all have been developed for a while. So economically, I'm talking about cost effect.
Cost, yeah.
Can you make some quick comments? Maybe one minute.
Cost, definitely EVs are more expensive than liposomes, for sure, because the liposomes are synthetic. But I think we're using large scale cell production facilities that we can gradually bring down the cost. But right now it's more expensive, for sure.
The second question, obviously, I think that there are many questions here touched upon this very interesting observation. EV derived from specific tissue types, tissue cell types that have higher kind of tendency going back to those tissues.
Yes, we call it homologous targeting.
Right, it's like a tropism.
Yeah, tropism. Yeah.
I'm wondering the mechanism. Really.
Mechanisms, I think it's something must be to do that the EV surface has something that it has a high affinity with the parental cell. So it will go back to the parental cell. So actually there's groups, including our group, using cancer-derived EV to deliver chemotherapies to the cancer. We see homologous tropism, that the EV will go back to the parental cell who produced them. But the mechanism, we think it's something to do with the binding molecule, but it's not 100% elucidated yet. Okay. We don't mess up with the agency, so.
I don't think there's a question in the Zoom board.
Yeah, there's nothing. Yeah,
So
right.
let's thank our speaker again. Thank you very much for being here. Thanks, Brad, for coming.